Forensic characteristics of semen. What is semen? Semen is the ejaculatory fluid from males. It's all of the fluid, with both the liquid portion and the sperm cells together. The spermatozoa, the sperm cells, are the male gametes. They're the male sex cells, and each sperm has 23 chromosomes. It has all of the chromosomes from the male, and it's got a mixed up combination of stuff that male got from his mom and his dad. Sperm are created in the testes. So you have a testis, and that's the epididymis, and it's carried away from the testis through a tube called the vas deferens. It goes into the body and loops around behind the urinary bladder, where it meets up with a gland called the prostate and the seminal vesicles. These add nutrients and sugars and extra fluid and stuff to the semen to help it through the process of ejaculation. From there, it exits the body through the urethra. Stains may be found, seminal stains, in a variety of locations, most commonly bedding and clothing. It can be difficult to distinguish stains that could be evidence versus stains not produced during a crime. For example, stains that you might find in somebody's bed sheets or in somebody's own boxer shorts. It dries quickly, it crusts stiffly, it can be very, very brittle. So if you have clothing or fabric that has a seminal stain on it and you bend it at all, it can flake off and fall off into dust very, very quickly. It can be difficult to locate because it is not very opaque when it dries, and it washes out very thoroughly. If somebody put their clothes through the wash, you can count it basically gone. The same three criteria are needed for semen as with blood. Is it semen? Is it human? And is it that person's semen? Three main tests are used for this purpose: acid phosphate test, microscopic examination, and PSA tests. The acid phosphate test uses exploits an enzyme secreted by the prostate into the seminal fluid. This acid phosphate is found in other body fluids, but not nearly as concentrated as in seminal fluid. It's something like 400 times more concentrated there than other bodily fluids. So what you do is you use a solution of sodium alpha naphthyl phosphate and fast blue. You moisten a filter paper, you rub it on the item that you suspect has a seminal stain, and then you apply this solution to the filter paper. You do not spray it directly to the evidence item. If it turns purple, then there is acid phosphate. Again, other fluids can have acid phosphate. Just because it turns purple doesn't necessarily mean seminal fluid. A very slow development of this color, in fact, implicates other types of fluids. Vaginal fluid, for example, can have acid phosphate in it too at higher concentrations than interstitial fluid or saliva. If the color change happens in less than 30 seconds or so, you should be pretty confident that what you're dealing with is in fact a seminal stain. The quicker the color change, the more definitive it is for semen. You can also add this chemical, I'm just going to say MUP, I can't pronounce the other word, to be added to it for fluorescence in case the color change might not be able to be apparent given your circumstances. Unfortunately, the acid phosphate test has a variety of false positives. It can come up positive with some vegetable and fruit juices, different kinds of funguses, which is ubiquitous in the environment. You have all kinds of molds and spores and stuff in any home. Contraceptive creams come up positive, and vaginal secretions also come up positive for the acid phosphate test. So it's pretty specific to semen. If it develops very quickly, you can be quite confident that it's semen, but this test is not truly exclusive to semen. You can, a popular way of doing this test is using something called Fatibus paper, and this can also help differentiate salivary stains from seminal stains and help show salivary stains in an area where they might also be semen. So what this has is a starch derivative embedded into the paper, which you can moisten and then swab the stain just like before. You spray your acid phosphate test onto that filter paper and both will show up. If there's saliva, it'll show up in this bluish color, and if there's semen, it'll show up in this pinkish purple color. You could also choose to examine for semen with a microscope, and this is pretty quick and straightforward process. Sperm have a super unique appearance. There is no other cell in the human body that has this long flagellum, this tail like this. So if you see something with a tail like that, it's going to be sperm. A very small amount of seminal evidence is applied to a droplet of water on the slide. If it's already been dried, you'll take a moment to mix that into the water droplet, and then you let the whole slide dry. Look at it through a microscope and look for sperm. And if you can see them, then it's a pretty definitive positive test for semen. It does not necessarily show you that it's human semen, although overwhelmingly, we're not going to be finding too much of any other species in somebody's home, for example. Unfortunately, a lot of seminal stains might not actually be able to, you might not actually be able to retrieve sperm from every seminal stain. First of all, they bind really tightly to fabrics and they get really tangled up. This tail gets interwoven with fabrics pretty easily. And then the second it dries, it's extremely brittle. You go to take that off of the fabric and it just falls to pieces. The cell doesn't live very long at all unless it's in really ideal conditions. It's very, very easily destroyed. In addition, not all males ejaculate many sperm or any. So sperm count can vary very wildly between different men, and some men just don't have any. For example, men who have had a vasectomy will still produce seminal fluid because all the stuff that produces seminal fluid is upstream from where they cut, but they won't have any sperm in that seminal fluid. You've severed the connection between the testis and the ejaculatory duct. Prostate-specific antigen is used where you have a positive anti-acid phosphate test, but you didn't find any sperm. So you're pretty sure that it's semen. It came up positive with that color change in less than 30 seconds, but with a microscopic examination, you didn't see any sperm. The PSA test is almost specific to prostatic secretions. It turns out to be present in tiny amounts in other bodily fluids, so it could potentially come up positive with other fluids, though it's not terrifically likely. It can be detected by monoclonal antibodies or by gel diffusion. So we talked about this process in the immunoassay and in forensic characterization of blood stains. It's the same type of situation. Either you apply antibodies that are designed to specifically agglutinate to prostatic-specific antigen, mix them in a test tube and see if you find that changed band, or you put them in gel agar, wait for diffusion to happen, and then see if a line forms between those two wells where agglutination happened after diffusion. As with blood, though, the only definitive test to determine that a semen came from an individual person is with DNA evidence. Everything else is presumptive, and no other individual test we've discussed so far can definitively rule in one person and exclude all others, except for DNA.